All right, so we talked about natural selection yesterday, so I'm not really going to talk through this much because we've already talked about it. But to kind of summarize from yesterday, we talked about the four tenets, which include the fact that, or the idea from Darwin, that with each generation there's more offspring than can actually survive, that there's variation in that generation, that there's going to be adaptation because the traits are going to be different and some are going to be more fit than others, more able to reproduce. And then speciation, the whole idea that if organisms either are isolated or have some sort of isolation, sympatric or allopatric, that they will begin to become different from each other and no longer want to or be able to reproduce with each other, which would make them part of different species, which is speciation. Survival of the fittest we talked about yesterday, so it's the whole idea that organisms that have the best traits are going to be able to survive, have favorable mating behaviors, be able to mate, and be able to reproduce and send their genes off to the next generation. Therefore, the next generation will be more like them and have those same traits. Now, the conditions could change, and so the next generation, different traits might be favorable. But whichever traits are favorable, that generation will be passed on to the next. Descent with modification, it's just kind of echoing the idea of that there's changes through the generation. So with each generation, the genes that are most favorable will be passed down. And so there will be changes between the generations depending on what the environmental factors are that make a trait favorable with each generation. What role does genetics and DNA play in this? So obviously our DNA codes for everything about us, codes for all of our proteins, all of our traits. And so these are going to be what make us have those traits that are either favorable or not, and we pass them off, or they are passed off via reproduction, which, as we know, is passing off haploid DNA. All right, and then there are lots of examples of favorable traits. Maybe it's a mating behavior, maybe it's a scent, maybe it's a territoriality. There's all different kinds of behaviors that make an organism fit. Maybe it's strength. If males have to fight for dominance, maybe it's strength in that fight. But with this particular example with male birds of paradise, there's an example or a picture of it, I'm sorry, to the right. You can see the long, colorful tail. It's not ideal for flying, for trying to flee from predators, or necessarily trying to blend in, but it is good for attracting mates. So the longer and more brightly colored the birds of paradise male tail is, the more females it will attract, and so it's going to be able to reproduce a lot if it has a really long colorful tail. And the males in the next generation are going to also have long colorful tails because they're going to get genes from males who have long colorful tails. Okay, let's see. All right, some evidence. So I don't know if this is in the same order as in your notes. I think it might be in a different order, but biochemically, so this is going to be the chemicals and how they're arranged into molecules, and how that's consistent amongst organisms. So organisms with the most similar genetic code are by evolutionists predicted to be the most similar to each other. So like we are more similar to, say, a dog than we would be to, say, a pine tree. Okay, so we all have DNA, but the more similar the DNA is between organisms, the more likely they are related or diverge from the same organism more closely. So for anatomy, there are a couple of different pieces of evidence that evolutionists use to support evolution using anatomy. One is homologous structures, which I'll show you on the next page, and the other is vestigial organs. I will show you that on the next page as well with some pictures. Paleontology also, so fossils and their dating are a huge piece of evidence that evolutionists use to support the theory of evolution over time as the origin of species. Okay, so this at the top, the whale fin, the frog leg, the horse leg, all those structures from these different organisms and the way the bones look on the inside, those are homologous structures, which means that the structure is similar, but the function is different. So the structure between a whale fin and a frog leg is similar. You can see phalanges, carpals, you can see ulnas, you see what resembles a radius or an ulna, you can see what resembles a humerus. And so the idea is that maybe these came from a common ancestor, according to the theory of evolution. So the difference in function, though, obviously they function differently. Frog legs are for leaping, whale fins are for moving through the water. Okay, so there's, from a creationist point of view, it's, well, God's pretty smart, and he knew that, that it would work well, just as well in a whale fin as it would in a frog. But according to evolutionists, this will actually support the theory of evolution, that maybe all of these organisms came from the same ancestor. Then we have vestigial organs, and I give a few here. I'm only going to talk about a couple of them. So, for example, the muscles that move our ears. I can't think of any reason why it's good to be able to move your ears now. So the whole idea is maybe there was at one point. Maybe there was an ancestor of ours that needed ear muscles, and so that's why we have ear muscles. And also canine teeth. We don't seem to need canine teeth all that much. So maybe at one point our ancestors did. Okay, with that theory. All right, speciation. So we know that that is the origin of a new species, and that's usually because of some sort of isolation or barrier, but with a gene pool barrier. It can also be because of polyploidy, which is when there's more than 2 chromosomes that code for the same trait. So we have 2 chromosomes that code for, we have 46 chromosomes, 23 from each parent, right? So like for pair 1, we have 2 chromosomes, 1 from mom, 1 from dad, coding for the same traits. Same thing, 3 pair 23. Well, with some organisms, they actually have 3 chromosomes, and that's generally going to be in plants, and that's going to contribute to diversity. Think about all the diversity that comes from meiosis of organisms with 2 chromosomes. Imagine how diverse it can be if there were 3 chromosomes to be separated. So polyploidy in those types of organisms would contribute to genetic diversity. Also gene pool barriers, which we actually already talked about during ecology, but sympatric would be the organisms are in the same place, but they just don't mate with each other for one reason or another. Allopatric would be they are actually physically separated from each other. So habitat isolation, there's some sort of a physical barrier. Temporal isolation, remember that one is they're not susceptible to reproduction at the same time. Hybrids, that's going to be like a mule, which is obviously infertile. And then behavioral isolation would be like they don't respond to the mating behaviors. Okay, adaptive radiation and divergent evolution kind of go together. So those are both talking about, so adaptive, we think of changing over time to accommodate some sort of environmental factor. Radiation is like moving out. And so adaptive radiation is like with the Galapagos finches, how they moved away from each other, and therefore their environment was slightly different, and therefore they became slightly different from each other. Divergent evolution is the same thing. So the organisms moved away from each other, therefore their circumstances are different, therefore different traits are favorable, therefore their next generations are going to be different between the different groups, depending on where they ended up. Convergent evolution is going to be the opposite of divergent evolution. That's like with Viceroy and monarch butterflies. They ended up looking very similar to each other, having very similar functions, very similar niches, but they live in different areas. And so they actually became very similar to each other, even though they're actually part of different species. So instead of becoming more different, they became more similar. Polymorphism, that's going to be when there's multiple different expressions of a trait. So like with peppered moth. So the story of the pepper moth is actually in your in detail. I typed it out in detail in your notes there. But so there were, and this has been pre-industrial revolution, but so there were dark moths, and then there were light moths, and the darker moth was a more darker and lighter moths were pretty favorable traits leading up to the Industrial Revolution. But after the Industrial Revolution, they both kind of stood out because the soot from the factories would land on the buildings in the area, and that would cause both black or white to kind of stand out. And so the trait that started to end up being very favorable would be if moths had speckles, and so they look like the one on the left in the picture here. So they're kind of white, but they have the black speckles, and so they looked like the speckled buildings that were covered in soot. And so that became a very favorable trait. And so instead of having lots and lots of dark or really light moths, all of a sudden there were tons and tons of peppered moths. And it's just because they were able to survive and reproduce, and so they kind of started to take over the population. And that was transient polymorphism. Peppered moths are no longer common because that's no longer the case. The mill buildings in London are no longer covered in soot. So all of the different types of moths are indeed now similar in ratio. Okay, so sickle cell anemia, so this is balanced polymorphism. So this is not transient, this is not changing. It's not like sickle cell came around and now it's gone. It seems to be here to stay. So with sickle cell, the three different versions of this trait are the homozygous recessive, which is someone who has sickle cell, heterozygous, which is someone who has the sickle cell gene but does not have the anemia or the negative effects, and then the individuals who are homozygous dominant, and so they have the normal blood cells. And that's been around for a long time, since the malaria outbreaks we've already talked about became very prevalent, and it continues to be a polymorphism. Okay. So we've already talked about phylogenetic trees. Remember that they relate to a common ancestor, and they're trying to take all these organisms and say, okay, here's the common ancestor. Now let's see what organisms are most like each other, what organisms are most like the ancestor. So the ones that are closest to the ancestor are most like the ancestor. So like a gibbon is more closely related to the primate ancestor in this particular phylogenetic tree than the human is. The chimpanzee is more like the ape than it is like the gibbon, because it's further from the gibbon on the tree. Again, reviewing this, so remember a cladogram. Okay, sorry I had to pause, there was an announcement. But a cladogram, remember, is connecting organisms by traits, and so the organism at the very top, the chimp and the mouse, they have all of those traits listed below. They have jaws, lungs, nails, mammary glands, fur. Okay, so they have all of those things. So whatever traits are below the organism must have all of those traits. Okay, how to make a cladogram, and we'll practice this in class. But you need to make a list of organisms, make a list of characteristics that they could have in common, and then make a table to where you can kind of check what matches up. So like with this one, a sponge, it has cells with flagella, but it doesn't have any symmetry, it doesn't have bilateral symmetry, it doesn't have any of these traits down here. A jellyfish is a little more complicated. It has cells with flagella, it has radial symmetry, but it doesn't have bilateral symmetry or anything below it. So kind of working your way in this direction, you start to move towards more and more complicated organisms, and we could definitely make a cladogram from this. Okay, we've already learned how to Hardy-Weinberg. So you guys need to answer the questions on the assignment on Moodle.